Next up is Noah Pardell from the University of Alberta. They'll be talking about facilitation of transmission of axonal branch points by nodal GABA receptors, simulations with a reconstructed proprioceptive axon model. All right, go ahead, Noah.
All right, thank you so much. I'm just going to share my screen, and I hope everyone can see the screen. So I will start. Yeah, thank you for coming to my talk. I'm talking about branch point failure and the rescue thereof using ionotropic GABA-A channels. I'll just move the slides. So we have a synapse, but a computational way of looking at the synapse is an input signal that gets modulated in some way to create an output signal. Now, one of the ways you can do this is presynaptically versus postsynaptically. So presynaptically, there's this thing called presynaptic inhibition. Classically, what this means is that the output signal is inhibited, but the modulation and why it's inhibited, the vesicular release is diminished, happens presynaptically. So if we look at the schematic on the bottom, we see this lovely action potential that has reached the terminal. And historically, we believe that there is these ionotropic GABA-A receptors that allow chloride to, oh no, my thing, there we go, that allow chloride to efflux from the terminal, producing a depolarization. This depolarization was thought to reduce the resistance and create a shunt. And this would lead to less vesicular release. The problem is these GABA-A ionotropic channels aren't actually located at the terminals. So what are they doing? They're actually located at the branch points. So here we have a schematic with the 1A proprioceptive axon in the top going through the dorsal columns and sends off an axon collateral that travels ventrally through the gray matter. If the ionotropic GABA-A channels are at the branch points, then what contacts them are these GABA interneurons, which are the last of a train of interneurons that produce this phenomenon known as primary afferent depolarization, or PAD. This PAD can be measured more dorsally, and that's historically it has always been measured. So what is this PAD doing? What are these ionotropic channels doing at the branch point? Well, immunohistochemically, if we look at the branch point with this neurobiotin-filled axon, we see a co-localization of the GABA-A channels and the voltage-gated sodium, the yellow and the white, respectively. In a more diagrammatic form, it can be illustrated like this. So what is it doing at the branch point? Well, for our model, we took morphometry from a serial electron microscopy study in 1991 and reconstructed it in the computer. On the bottom is a electrical circuit diagram that corresponds to the double cable structure that we created. And this branch point highlighted here is the one that is used in the study, as well as the one that will be used in the next couple slides. So keep in mind this branch point. So to kind of validate the model, we had this proprioceptive afferent that has been filled with neurobiotin in experimental mice, and we see the branching structure, primary, secondary, tertiary branches. We wanted to see if the model and the experiment had kind of the same diameters, et cetera, for these different classes of branches. And so, as you can see, the secondary and the tertiary branches are quite concordant in their diameters, and this would help validate the model against experimental observations. However, there's kind of a discrepancy between the primary branch. There's some reasons for this, one of which might have been that the original electron microscopy study cut a little bit more ventrally than we thought, and therefore it's actually a secondary branch. But there could be some other explanations as well. Lastly, another way that we validated the model is through pairing the action potential heights to make sure that they were somewhat close to the experimental data. And here's just a model trace. So with the ion channels in the model, we titrated the conductance of potassium and sodium over a wide parameter space to try to see how common branch point failure is. In the black sections, this means that an impulse was not even able to conduct on the axon, and the magenta sections mean that there was unphysiological behavior. It's too hyperexcitable. So this region that's bounded by this trace is the one where there was impulse conduction that reached that branch point I talked about earlier, but either in gray, it wasn't able to go into either daughter branch, so the red and the blue were not able to conduct. In blue or red, it means that one of the daughter branches was able to conduct and the other failed. In green means both were successfully permeable to propagation. So in that parameter space, the gray, the colorful one, branch point failure is common, as we see in the pie chart. So if we increase chloride conductance with these GABA-A channels at the branch point, we should increase failure. So if we fix potassium conductance on the y-axis, and put it on the chart on the right, and on our y-axis, we titrate GABA conductance or chloride conductance, we would expect increased failure. We would expect the cells to go from green to blue or red to gray. But actually what we see is gray to blue and blue to green. This is an increase in, a rescue of the previous failure. It's increasing transmission. So actually presynaptic facilitation is what we're seeing. So lastly, we have an impulse which travels down the axon, is not able to go into one branch. But with the addition of PAD and these ionotropic GABA receptors, we're able to successfully go through both branches. Thank you so much.
Thank you so much for that, Noah. Great talk. All right, so again, we'll sit and we'll wait for the Q&A. Feel free to either send in your questions in the Q&A, or you can come on screen. I'll also monitor the chat for any questions that might pop up there.
Sure.
All right, so we have a question for you, Noah. This is from Gregory Piercy. He says, very interesting talk, first off, I agree. Does this suggest that presynaptic facilitation may be required for normal behavior and that a lack thereof might explain dysfunction?
Sure. So what we're thinking in the lab is that this presynaptic facilitation is being implicated in the synaptic priming of reflexes. So if there's branch point failure, that means that there's some sections of the collateral that are silent in everyday functioning. But if we're able to facilitate the impulse to go through that branch, for example, in a reflex, when you need to activate certain muscles that were previously quiescent to, let's say, I don't know, on the train to stabilize yourself when the train shifts, then that's an example of a normal behavior that we think that this is acting as, the facilitation is doing. A lack thereof very well might explain some dysfunction or some areflexive states. I'm not too sure about the clinical side of if this priming wasn't there, but that would be my answer to that question at the moment.
Thank you. Yeah, I think that definitely answers that. All right, we are just gonna hang out here and wait for some more questions to come in on the Q&A or in the chat box. Again, you can raise your hand and we'll bring you on screen, but we'll just sit here and because these are the longer question and answer sessions. All right, great. We have a follow-up from Gregory. Would this help to explain a cutaneous facilitation of reflex amplitudes that was once thought to be a reduced presynaptic inhibition?
So actually cutaneous fibers are, for example, I wonder if this is still sharing, correct?
Yeah, perfect.
So where are we? Here we go. So as we see here, the 1A afferent here is in gray, is contacting the interneurons, which then contact the branch point. And this is the classical PAD story. Cutaneous afferents are actually also an area that can activate this circuit and create, as you say, a reduced presynaptic inhibition. What we think is actually this facilitation, this increase in conduction through the previously quiescent branches to increase the postsynaptic response because there's just more transmitter across all the branches. So I would say that this helps explain that, yeah. I could have added other little circles here to show, other, central nervous system, activations on this circuit, but I wanted to try to keep it a little simple. But cutaneous afferents are definitely ones that produce this tonic pad that we see this facilitation acting. Yeah, for sure.
Yeah, it could definitely get complicated very quickly.
Yeah.
All right. Thank you, Gregory, for those great questions.